Let us go next. Next topic in our syllabus is the RNA processing. RNA processing means what? We have seen that transcription takes place, that is from DNA RNA molecule is made, and this RNA has got multiple varieties. You have seen it earlier during when you have studied physiology, that we have got varieties, that is messenger RNA, transfer RNA, ribosomal RNA. There are varieties of RNAs. Now how it is processed and what are their significances? So the first thing is, the immediate thing is why we are calling it as a post-transcriptional modification, or people call it as a post-transcriptional modification. We will just discuss it shortly. Basically, whenever there is a transcription, that means what is done is from DNA, that whatsoever coding is there, whatsoever information is stored in the form of codons, that is converted into another form, which has been easy for the other material, cellular material, example ribosomes, to make it a functional product, that is a protein. So that is just like a computer, that we put something that is taken by computer into its own language, that is we call it as a binary language or 0-1, like that. So it takes in that whatsoever we give input, that is taken into in its own form, that is the transcription computer is doing. And when you want it in any form, may it be audio, may it be, again it is converted into a different form. So this is how the process takes place. So here also there is a post-translational modification is there, but let us see what happens to RNA. So first is the RNA is made, that is first RNA is made, that is the messenger RNA. That messenger RNA will be transcribed into a mature messenger RNA that will be capable of being translated to a protein, and this protein includes a major step, that is significantly making changes in the RNA molecule, the addition of 5' cap, the addition of 3' polyadenylated tail, and RNA splicing. I am very sure, if you remember, if you have seen the tortoise, splicing is very important feature, it has been there, and this process is vital for the correct translation of the genomes, because the initial precursor mRNA produced by transcription often contains both exon as well as intron. Exon means the area which are required, which are for coding, while the introns are there which are non-coding sequence, but they are coming in the flow because that DNA is a long one and it is coming. So splicing removes this the introns and it links exons directly, so that the protein comes in a continuous feature, whatsoever protein is required, and that structure is maintained. So let us see how it happens. This post-transcription modification may also occur during the processing of other transcript, like transfer RNA or ribosomal RNA or any other type of RNA which is used by the cell. So these are a variety of such RNAs which are there, and these RNAs are processed, they are changed, they are structurally modified, and accordingly it is there. Let us see how this messenger RNA is processed. So we have seen so far the process or the controllers or regulator. So there is a DNA molecule, and on this DNA, this is a DNA molecule, there are various regions which are there, that is enhancer or silencer region is there. Enhancer region means from where when this we have seen earlier transcription factors or having a DNA binding domain. So these are the regions where bound by various protein. We have seen this protein. What are these? Transcription factors or activators. Okay, and this is the they have DNA binding domain, and here there is a DNA. So on this particular part they will bind. Then they will have the sequences, that is proximal and core promoter places, that is on the DNA, and from that there will be promotion. Then the next area on the DNA is there, that is called as a 5' untranscribed region, the region which is not transcribed for anything. Okay, that has no important information. And then next will be there, that is a start codon, which is very commonly known as a start codon, and it is the first codon for the messenger RNA transcript from the parent DNA strand. So that will be the start codon. In most cells it will be there, that start codon will be there. Then next will be the open reading frame, that will be the DNA, it will be part of DNA, and in that this much area which has to be read or which has to be transcribed, that area is the fixed one, and this open reading frame will be there, and in that area it will be there. At the end there will be a stop codon, then that next will be the 3' untranscribed region, and this 3' in that there will be a terminated codon will be there. So where our transcription factors, which are silencers, okay, they will be there. Here once it comes to the terminated codon, then these factors will be binding to the particularly this one or repressor portion, we can call it as a repressor also, and here it will be stopping the transcription. This is what the process of transcription. So what happens in that, the next thing is this is what the thing which has been found in the, or which has been synthesized or formed during the transcription, that is whatsoever portion of DNA there from the 5' untranscribed region to 3' untranscribed region, that much whole area has been formed. But as I told you, it has got all sections, that is what, it has got exon and it has got intron. Intron are the, we have discussed this, intron is the nucleotide sequence within a gene, and that has to be removed by the RNA during splicing, because this area does not code for protein or amino acid sequences, and it has to be removed. Now this is these introns are there. These introns are done by various splicing enzymes. There are a variety of splicing enzymes are there. This splicing enzyme will cause breaking here, and this breaking will result into, or that much chopping is there, that much portion will be chopped. Then again next portion is of exon, then again there is a intron, this intron again has to be chopped, and then again there is a exon. So what this splicing process will do is, that will chop off these introns, this intron and this intron will be chopped off, and then we will get a continuous sequence of exon. So this is what exon. So this exon, then the middle exon and the last exon, and there are two ends, that is 5' cap and poly A, that is polyadenylated tail. So this poly tail, that will contain multiple adenosine monophosphatase, and it will be stretch of RNA that has only adenine bases, and it is process that produce mature messenger RNA. So this is the last portion that will be carried to the ribosome, and then ribosome will make or will do the translation, and it will cause the protein synthesis. Now it is very, very important into this, that how this happens. There are multiple varieties, the thing is same, but only thing is there is a example is, one more example of it is, that is called as a polycistronic operon. Now operon means what? It is the area where a similar transcription factor, that is the transcription factor is working on that particular place, that is called as a operon. Okay. Now this operon, if there are multiple in nature, so it is a functioning unit of DNA, this operon is, which containing a cluster of genes under control of single promoter. We have seen variety of transcription factors, and there are promoters. So if they have multiple, single promoter is there on DNA, promoter moiety is there, and there are multiple genes are there, which are multiple in nature. So they are transcribed together into a messenger RNA strand, and they are either translated together in the cytoplasm, or they undergo splicing to create monocistronic one. So now here suppose there are 10 proteins they are put in one operon. Okay. Now, if you require only one protein, then whatsoever 10 this messenger RNA which is formed for 10 one, now it will be spliced and the 9 will be put aside and only one which is there, that will be made for protein synthesis. So, this is how these operands are working. In this operon, the important thing is it can be, as I told you, it can be single only in certain places, or many places it can be multiple one. So, when it is multiple one, we call it as a polycystonic operon. When it is single one, it is cystonic operon. So, because of the splicing, we can have, see, these are the operon open reading frame here, that is for second one, it is open reading frame, and we have found these are various RNAs, and from this RNAs, this first protein coding region, then second protein coding region, and maybe many more, as I told you 10, here the example is given for 2. So, this for 2 it is there, so this has given one protein, this is given another protein. So, this is what we call it as a polycystonic operon. Okay. Now, this one, it is having the ribosomal binding site initially. Okay. Here also it will be ribosomal binding site, and from this ribosomal binding site, the next process will be there, that is coding for particular protein. So, now this, when it will be translation, we shall be seeing later. So, this protein will be formed according to this. So, the pre-mRNA molecules, they will undergo 3 main modification. One, these are what? First is 5' capping, then 3' tail, that is polyadenylation and RNA splicing. And these all 3 things occurs in cell nucleus before the RNA is translated. The RNA translation most of the time, except it is the RNA which is made for the nuclear factors or other things, in most of the time it will be this messenger RNA will come out, it will bind to the ribosomes, and then the protein will be synthesized. Okay. In rare occasions, this RNA can give rise to protein inside the nucleus itself, in very less cases. So, first thing is the 5' processing. It will, because of the addition of 7-methylguanosine structure at the 5' with the various enzymes like phosphatases, etc., and these all enzymes will cause the attachment to this, and thus it will produce the cap on that. This cap will give the marking, okay, that this is the initial portion, so that it will be accordingly transcribed. The cap also protect the 5' end of the primary RNA transcript from the attack of ribonuclease enzymes that can specificity to 3' 5' phosphodiesterase bond and phosphodiester bond, and because of that this RNA will not be damaged. Then the next one is the 3' processing, that is we call it as a polyadenylation, or the provision of the tail at the end. So, this tail will be there, around 250 adenine residues will be attached to form the tail, that is adenosine tail, and this cleavage and adenylation reactions occur primarily if the polyadenylation signal sequence is located near 3' end of the pre-RNA molecule. Now, this there can be one more sequence, that is guanine and uracil rich sequence can also be there, and it usually present for the downstream or pre-mRNA molecule. More recently, it has been demonstrated that the signal transition sequences like UG, UA upstream of the cleavage site can also direct cleavage and polyadenylation in the absence of these signals. So, these are how these things are being done. These are important particularly for transfer of sequence specific binding protein for cleavage and polyadenylation. Specificity factors are there, cleavage factors are there, then cleavage stimulation factors are there. They will all working with the RNA polymerase enzyme, and accordingly they will work for various aspects like stopping the activities and then making the breakages when required and stopping the final one, and again protection of the end of the RNA molecule. So these are various activities they are producing. Then splicing is really a very interesting one, that you can understand the specificity that only few areas are removed, that is the introns are removed which are not coding for protein, they are removed, and only exons are there, and these exons which are there, which are between some introns, whatsoever exon was there, so all the exon parts they are connected together to form a single continuous molecule, and these are the sections which become expressed or translated into protein. So they are the very important, they are actual the coding protein of the messenger RNA molecule. Most RNA splicing occur after the complete synthesis and end capping of the end capping we have seen, that is polyadenylysine. So this happens, transcript may with many exon can also split simultaneously, means during transcription only it can happen, before the because capping happens only after end, but before end only splicing can be there. The splicing is done by a large protein complex that is called as a plysiosome, that is assembled from proteins and small nuclear RNAs, and they recognize the splice sites in the pre-mRNA sequences. Many pre-mRNAs they include antibodies. They can be spliced in multiple ways to produce different mature mRNAs that will encode different protein sequences, and this process is known as a alternative splicing. This allows production of large variety of proteins from a limited amount of DNA. So DNA for every type of protein, example, as if you know, let me explain you again in clear way, that every time new bacteria, new virus or something new is there, or new foreign object comes, new antibody is formed. Okay, so that information has to be coming, because protein synthesis, any kind of protein, antibodies are the proteins, and any kind of protein has to be made from the genetic material. So for this genetic material, for these antibodies, this information goes to the DNA genetics. Accordingly they will have the sets, and from that RNA will be made. Now for every so many antibodies, how many DNAs we can have, isn't it? So accordingly the DNA will be limited, but from certain areas the RNA will be made, and as per the required structure of antibodies the RNA will be spliced so that that particular structure of antibodies can be made. So this type of process is there, that is called as alternative splicing. One more process is there, we have seen these three processes, that is histone mRNA processing. That is, we have got histone is a protein, and that processing, as I told you, it takes place in nucleus itself. Now what is interesting in this is, or what is important in all this process is, this process is very important, particularly when you will go in 5th year or whenever the word comes, that is pharmacogenetics, you will find that at many places there are changes. Okay, pharmacogenetics is a whole branch which is devoted for this, and because of this genetic differences, what can be the problems? So there are many problems so far has been reported, because you can imagine, you are a student of pharmacology, you are studying pharmacology since last 2 years, it's last year and almost this year. So here you must be knowing what is the situation. Situation is what, that this all, you know, everything is, every protein or almost all activities, may it be enzyme, may it be structural, or may it be functional protein, most of these are, very few exception of other molecules like lipid or carbohydrate, but most of these or majority of these are the proteins is first thing. All these proteins, even for making carbohydrate or for making lipid or for metabolism of this, they are proteins, because they are all enzymes, because without enzyme it will not be made. So that means proteins are very important. Any change in the protein, that will affect. Now you have the list of proteins. Okay, this if it is structural protein, they can be receptors. If functional protein, they can be enzyme, anything like there. Now think about, imagine about splicing. Now if there can be a simple small change of one nucleotide, because it is a sequence, okay, and in RNA it is AUGC, isn't it? Instead of thymine we have got uracil. So in case of that, and this sequence is there, we know that when they are in a 3 or in a sequence, then we call it as a one codon. So if the sequence, if the splicing changes, if the splicing accuracy is just changing by one nucleotide, you can imagine, suppose here here the set of codon is there, that is AAUAA. Now before splicing it is correct splicing, but suppose it comes after A, then if the splicing enzymes make a mirror, that is after A it becomes, then whole codon sequence change. If it whole codon sequence change, then whole protein formation will be changed, because its structure will be different, different kind of amino acid will be put into it, and because of that the protein which has formed for particular function will have totally different structure, and because of that it may work less or it may work. This type of proteins, they are called as a splice variants. So you will learn about this, you will have this term many times, because in pharmacogenetics this word will come many times, that because of change in single nucleotide, okay, that is known as SNAPs, single nucleotide addition or deletions proteins are there. So this protein, because of change in the single nucleotide, it may be because of splicing, it may be because of any error that is happening at the DNA level or transcription or translation level, wherever the error may be, that will result into problem in the protein, and that can changes at many places. Actually it started with the term that is called as a idiosyncrasy, then it was later put into pharmacogenetics that idiosyncrasy was happening because of actually different in the metabolism rates. In some people the rate of metabolism is higher, in some people it is metabolism is lower. You must be aware, or you might have encountered various things. Example, in paracetamol and in other places we call them as a slow acetylator, fast acetylator. In tuberculosis also you will encounter this. So this type of molecules are there which are having slow or fast acetylation activities, or slow or fast metabolism activity. During last year also metabolism or biotransformation, you might have studied this. So these particular things, they are mattering much. So therefore in some people the metabolism will be faster, in some people metabolism will be slower. Where metabolism is slower, there is the accumulation of the drug. Where metabolism is fast, there is a what we can say stoppage or removal of the action, and this all may affect the patient depending on the case it may be. So this is how this variant, splice varies and our protein variants are important. So I hope you understand this, what is splicing, all these terms. If you wish, you can go for more detail, but since we have to cover so many things, after that we'll just give the glimpse. I am not going into more details for each topic, only what's prescribed in the syllabus, and we are trying to put it to it. Now already you know about variety of RNAs. I expect that you go through it. What are the mRNA, what are the tRNA, what are the rRNAs, and how they are functioning. Okay, you have seen it. Next time, if possible, we will make it very small glimpse, or rather we will go directly for the another topic. If you need it, then we may go for it. But I expect you go through various things, you study the things, and if you have any difficulties, you can ask. because this is as I told you earlier, this is a very emerging area now, which has been merging the genetics into pharmacology. Earlier the genetics was not that much merged with the pharmacology, and pharmacology was only concerned with the drugs. Even the classical pharmacology so far we studied or we taught, it never had this. But this is really interesting, because this gives you idea about why there can be a problem, and what are the various controlling mechanisms which are the basis for the problem. So far in pathology you have studied that what are the structural functional things which are happening, but you never studied the what are the reasons behind these structural or functional changes. Okay, here you study that. Suppose there is atherosclerosis or there is a cancer, you say that this happens, there is a growth of things, there is a so and so are involved, but why this? So for still what we call it as a molecular or genetic basis of all the problems or most of the problem we study here, and therefore wherever possible most of the drugs are being made into these areas. Maybe in next 5 to years whole pharmacology will change, because the most of the drugs which are having lesser specificity, and if the specificity is less, then adverse effects are more comparatively, so will be tried onto that, and maybe so many drugs will be there which you can see. Example, we have recently seen in what we say about this immunology, that how the new monoclonal antibodies and all are available, which were not earlier available. Earlier immunology or immunotherapy was very small topic, comprising maximum 3-4 classes of the drug. Now we have got so many drugs, and henceforth this will be happening. So this is how the things are there. We shall go for the next topic whenever we get time accordingly. Okay. If you have any difficulty, I already put many a times, I am really unhappy with your class that attendance is not good. Hardly 11, 12, 13 students are there, and already I have put it very clearly on the group, and you can put it again, particularly you can inform your friends that I have a habit to ask whatsoever I have taught, and जस्ट सर, गुड आफ्टरनून. एक 5 मिनट में मैं कॉल बैक करता हूँ सर आपको, 5 मिनट में करता हूँ. So this is how these things are working. I have a habit that whatsoever I teach, I can ask from it. If you have studied, it's fine. If you not, I cannot help you in that matter. Yes, I can help only in what situation is whatsoever things you have having difficulty. If you are going through it, if you ask difficulty, I am definitely there to help you, and you can make the session whatsoever you want about it. You go through the things, you can ask the difficulties, and you inform everybody like that. Okay, so please make it clear. Please see that you all are doing it, and if you have any difficulties, please ask definitely. And if you study it, if you try to make the questions, you will understand how the questions are made. Okay, so try to do it. If not, then if there are no difficulties, it's good. Okay, I am happy that my efforts are appreciated by you people. So thank you again. Take care. And if you have any difficulty, please definitely ask me.